Hello everyone, my name is Kephas Mphande. Welcome to another video. Today I'll be talking about what I do in the lab. So I've been working on developing a pathogenicity test for Serratia marcescens. It is a bacterium which causes Cucurbit yellow vine disease, known as CYVD. This bacterium was discovered in the early 90s in Georgia and Oklahoma. It was found that this bacterium is transmitted by squash bugs in the field. The typical symptoms for CYVD include bright yellowing of the plant, phloem browning, stunting of the plant, and plant collapse. These symptoms usually occur when the plant has reached reproductive stage and is putting on fruits. Therefore, it has been a challenge to come up with a pathogenicity test which can capture these symptoms in the lab, because it takes much longer for you to see the symptoms. In my study, I'm looking at developing a pathogenicity test which is robust, which is also reliable, and can give rapid results. I have been investigating the effects of pot size and also the types of symptoms which develop in the growth chamber when you inoculate Serratia marcescens on squash. So what I do in my method is that I plant squash seed in 3 pot sizes: 4.5 inch size in diameter, 6 inch size, and 7 inch size. 7 days after planting, which is approximately at full cotyledon stage, I inoculate my plant with Serratia marcescens. So how I do my inoculation: I use a florist frog pin to make wounds on the cotyledon of the plant. Then I pipette 200 microliters of bacterium suspension onto the wound, or I also inoculate phosphate buffer which acts as a control. Immediately after inoculation, I start observing the plant for development of any symptoms. I basically look at 2 things which tell me plant growth rate. These include the leaf area and also plant biomass. I also use a spad meter which measures the quantum yield, which is a proxy for photosynthetic activity in the plant. So these parameters tell me how much a plant is growing, or the growth rate of the plant. If a plant is sick and is not growing well, I expect the biomass to be lower and also the photosynthetic activity to be lower. At 2 weeks after inoculation, I harvest my plant and take the dry shoot biomass. Also, from the stems of the plant, I extract the bacteria from the crown area and grow this bacteria on LB media. I characterize the colonies which grow on the LB plates and isolate those which look like Serratia marcescens. Then from the colonies which look like Serratia marcescens, I do PCR on them to identify whether what is causing plant sickness is CYVD or not. Thank you very much for watching. We'll keep you posted about what we get from our experiments. Thank you.